see just the GFP expression in the mesenchymal stem cells without gene jammer and with gene jammer. And we did do flow on this as well to show this. When we did use the gene jammer, we were concerned again about the possibility of detrimental effects and toxic effects on the cells. We did not see any obvious toxicity problems when we looked at PI positive cells. So these were not dead cells that we were seeing the GFP in. We did a number of experiments over time looking at the effect of the adenoviral dose on the transduction efficiency. So increasing adenoviral concentrations, obviously without the gene jammer, you saw a steady increase in GFP positive cells, but again we plateaued fairly early with the gene jammer. So this is obviously important that you can use lower doses of the adenoviral vector and get still very high transduction efficiencies. So again, this is just a relative fluorescence units. Again, some indication of the level of or number of viral particles entering the cells, and remain fairly constant without the gene jammer, but at the very high end of gene jammer, you can really load up the cells with the vector. And again, we did not see any really statistically significant detrimental effects when using the gene jammer. When we're very interested in because different culture conditions can affect the differentiation of these mesenchymal stem cells, we're really interested in looking at the effect of fetal bovine serum concentration on the transduction of the mesenchymal stem cells. And even at 0% fetal bovine serum, we still saw a relatively high level of GFP expression, positive cells, and we could also go up all the way to 20% fetal bovine serum and still get good transduction with these vectors. So from 0 to 20%, we're still relatively uniform in the transduction efficiency. So we're quite happy with those results as well, because again, if you're going to use this in a cell that you're going to look at differentiating them later, if you want to use with different concentrations of fetal bovine serum, or hopefully without fetal bovine serum in a more defined system. The next experiment, we looked at transduction of the mesenchymal stem cells, and now this just with with just the 85 alone fiber. So again, we moved back to just the more primitive type of adenoviral vector, and again, it did not seem to matter in the gene jammer transduction that whether it was the 85 or the 85F35 vector, we still saw very high levels of GFP positive cells with that, and again, no detrimental effects on viability of the cells. And again, you can see the different amounts of GFP expression in those cells. Okay, we did look at lipofectamine as a transduction efficiency as well, and yes, we saw some beneficial effects of the lipofectamine very similar to the gene jammer in many ways. However, we saw with lipofectamine there was some detrimental effects on cell viability with using some of these concentrations. So higher ends of lipofectamine, we did see some problems with the viability of the cells. When we looked at differentiation of these cells transfected with the GFP, we did see that the cells, again, could differentiate even though they had the adenoviral vector transduced into them, which isn't surprising. And so, again, with the oil red, we saw positive cells as well. So these cells could differentiate again in the chondrogenic and osteogenic pathways even after transduction with these viral vectors. And at relatively high rates, again, which was very encouraging as we move forward with this work. So with the first study of conclusions, the porcine mesenchymal stem cells are poorly transduced with just the adenoviral vectors, but the gene jammer transfection had a marked increase in transduction without any toxic effects. And it's not restricted to one type of vector. It had similar results with the 85 and the 85F35 vectors. Even with the addition of approximately 10% serum during transduction, there was some improved efficiency. But again, the main thing was that you could vary the levels of serum and still get good rates. And they do differentiate into the multiple pathways. So the second study, we were looking at the effect of gene jammer, not on pig mesenchymal stem cells, but this time with human mesenchymal stem cells in rodent cell lines. And objective of this, and this was done in collaboration with a group at Baylor Medical School, to investigate the effects of gene jammer on the efficiency of human mesenchymal stem cells with adenovirus containing GFP. Again, as if the gene jammer's positive effect on transduction was dependent on the presence of the receptors. And we believe that it was an independent method, but we needed to check that out for sure. And then to investigate the effects of gene jammer on the transduction efficiency of human and murine cell lines with BMP2. So similar in many ways, we looked at different viral particle loading of the cells, and again we saw very similar effects with the human mesenchymal stem cells that without the gene jammer is fairly low at the low concentrations. If you obviously, as you increase viral particle concentration, you get a higher transduction efficiency, but again, you were able to get, actually this is reversed, sorry. The gene jammer is in the white bars, and the control is in the dark. So we did see very similar results as we had seen with the pig cells as well. And you obviously, if you look at flow cytometry for quantification, this backed up the other results we see. So the next experiment in this group of studies, we're asking, does gene jammer enhance uptake through adenoviral receptors? And so looking at various cell lines with different receptor populations, the A549 human mesenchymal stem cells, CHO cells. So, and we looked at different viral vectors and with and without gene jammer. And the endpoint was, again, flow flow to look at the percentage of cells. So if you look at the 549 cells, high levels of the CAR receptor, the integrin, as well as the CD46, whereas you see with human mesenchymal stem cells or bone marrow-derived mesenchymal stem cells, very low levels of the CAR receptor, higher levels of the integrin and CD46, and it shows cells has low all the way across the board. So we had a varied number of cells with different characteristics as far as their cell surface receptors. With the 549 cells, there was no difference between the two vectors, or there was no difference between